Okay, so we'll just wait for 1 minute and then we'll start the class. Okay, so we'll start the class if yeah. So we have already completed our syllabus, but as I said in the last class, just a minute, yeah. So as I said in the last class, that in this week's class we will be uh, uh, looking the translation in eukaryotes in detail, and we will also be solving some of the questions related to translation in eukaryotes. So that's what we are going to do, because last class we could not complete translation properly. So I thought that this week we'll focus on that. And then probably on next, not next week, probably on 18th, today 16th now. So on 18th, I'll take a 1 hour class, and that would just be a doubt clearing session where you can ask your doubts, and also I'll be discussing we'll be solving some of the questions, like more questions from whole of the syllabus. So today, like we'll be discussing about translation, I'll be teaching about translation, and then your syllabus would be over. And then in the last class, which will be on 18th, on 18th from 7 to 8. Okay, 1 hour class, it would be a 1 hour class. And in that class, we will be just trying to quickly revise what all we have studied, and we'll try to solve some questions. So mostly we'll focus on questions in that class rather than on theory, because theory in detail we have already covered. So we'll be discussing the questions in that class. Okay, so yeah. So I'm just writing it in the message also, that 18th 18th April from 7 to 8 p.m. Okay. So this would be our last class. That is day after tomorrow. And in this class, whatever doubts you have, that we'll be dealing, and plus some questions that from the whole syllabus. Okay, so now I will. Sorry, uh, anybody has to ask anything? Okay, then I'll start the class. Okay, fine. So as discussed earlier, that we will be studying about translation in eukaryotes. So we have already dealt about translation, and I'm sure that in yesterday's class also you have had a fair a description of what translation in eukaryote is. But today we'll just try to read it in a detail, as in we'll read it in a more systematic manner so that it becomes clear, and then we'll try to solve some questions. So if we talk about what is translation in eukaryotes? So what is translation? It is the mechanism by which protein is synthesized from the information coded on the molecule of mRNA. That means, just a minute, I think someone wants to join. Yes. So I'm just repeating for those who have joined. So in the last lecture, we could not, yeah. So in the last lecture, we could not thoroughly learn about translation because of time constraints. So today we'll give our whole time for translation, because translation is a very important topic for your this exam point of view also, and in general for your curriculum, for your course examinations, or if you are going to give any competitive examinations, in that also translation is important. And especially why we are dealing with eukaryotic translation is because prokaryotic translation is easier, as in eukaryotic translation is a little complex because it involves more number of factors, transcription factors, and the mechanism is slightly a little different. So when we study about translation, by default we study about prokaryotic translation, so we do not get a much idea about how eukaryotic translation occurs. That is what we are going to deal today. Fine. So basically, translation is, we are translating. What we are translating? We are translating the information which is present on the mRNA. Now, when I'm saying that translation is actually the translation or translating the information which is present on the RNA. That means the information in the RNA is read and it is converted to amino acids. Now, how the information on mRNA is present? It is present in the form of ATGC, and which we call as codons. So information on the mRNA is present in the form of codons. And this information is read and it is converted into proteins or amino acids. Okay. So now, if we talk about eukaryotic translation, here we are only talking about eukaryotic translation. Okay. Just a minute. Yeah. Fine. So here we are going to talk about eukaryotic translation. So what are the components of a eukaryotic translation machinery? A eukaryotic translation involves mRNA, messenger RNA. What is the function of mRNA? It contains the DNA sequence or the genetic information in the form of codons to form the proteins. Okay, fine. Then we have tRNAs. What is the function of tRNA? It behaves as an adapter molecule between amino acids and or codons. Okay, and it is required to carry amino acid to the ribosome. That means, so proteins are synthesized in the ribosomes, but different amino acids which constitutes the protein, it has to come to the ribosome in order so that they can form a peptide bond and they can form a chain. So tRNA is the one which brings the amino acid on the ribosome. Okay, so ribosome is responsible for the formation of peptide bond. Means in ribosome, peptide bond formation is happening. But for that peptide bond to form, individual different amino acids has to come to the ribosome. And those individual different amino acids are brought to the ribosome with the help of tRNA. Okay, so what is a protein? Protein is a polymer of, protein is made up of what? Tell me. Protein is made up of
Amino acids.
amino acids. So proteins are made up of amino acids. That means individual amino acids are joined together by a peptide bond and the whole chain is known as a protein. Individual each and every component is known as amino acid, and the whole chain, whole peptide, whole polymer is known as a protein. So this is the function of tRNA. Then we come to ribosomes. So ribosomes are the protein synthesis machinery. It also has RNA which helps in translation. So basically the actual synthesis of the proteins. So when we talk about the protein synthesis, that does not means that First amino acids are getting synthesized, and then amino acids, individual amino acids are already synthesized. They just come to ribosome and they form a chain. So protein synthesis machinery, if I talk about, or protein synthesis, when I talk about, it is just the joining of the different individual amino acids. Okay, then we have different factors like initiation factors, elongation factor, termination factors. Okay, and then some enzymes are also required or involved in the translation, and these enzymes are involved in two of the processes, and these are the two major enzymes which are required. So this is important. So one is amino acyl tRNA synthetase, and another one is peptidyl transferase. So amino acyl tRNA synthetase means it catalyzes the attachment of tRNA molecule to its respective amino acid. That means if this is your tRNA molecule, suppose this is your tRNA molecule, this is the 3' end and this is the 5' end. Your amino acid, it always joins at the 3' end. Okay, and this is 3' end because it accepts the amino acid. Therefore, this is known as acceptor R. Now, this amino acid binds to the tRNA. This is an energetically expensive process. That means for the joining of this particular amino acid to the tRNA, ATP is involved. First thing, ATP is involved, and because it is an energetically expensive process, that means some enzyme is there which catalyze the reaction. So what can anybody tell me in general what is the function of an enzyme? What is the function of enzyme? What enzyme do? Anybody? What is the basic function of an enzyme?
Speeds up the reaction.
It speeds up the reaction. Exactly. That means enhance the rate of reaction. Correct, Madhavi, you're absolutely correct. That means reaction will occur without enzyme also. But when you're providing enzyme, it enhances or it catalyzes the reaction, and the reaction takes place very fast. So this attachment of tRNA, amino acid to the tRNA is catalyzed by an enzyme which is known as amino acyl tRNA synthetase. And the formation of peptide bond between two individual amino acids, this peptide bond formation, this is catalyzed by peptidyl transferase. Okay, it catalyzes the sequential transfer of amino acid to the growing chain. So these are some of the components that are required in a eukaryotic translation machinery. Fine. Now this is what has been told in the previous class also, because we are studying RNA biology course, so we should know that obviously everything is connected. But all the three kinds of RNA, whether we talk about mRNA or tRNA and rRNA. So all the three kinds of RNAs have a role in translation. They all are required in translation. So mRNA, how mRNA is required? Because mRNA contains the code. mRNA has the sequence of amino acids that has to be added. So if ATG codes for methionine, so methionine will come in the protein or not, it would be decided by the code, the sequence of the mRNA. So mRNA is important because it is coding. It is coding the sequence of the proteins, the sequence of amino acids. That means in a protein, whatever amino acid 1, amino acid 2, amino acid 3, amino acid 4, and so on. Suppose it is a 25 amino acid sequence. So whatever amino acid has to be added, suppose it is methionine, it is arginine, lysine, glycine, valine, alanine, whatever it is. So whatever amino acid has to be added in the protein, this sequence is decided by the mRNA, because mRNA has this sequence in the form of codon. Fine. So that is why mRNA becomes so important, and that is why mRNA is known as a coding RNA, and tRNA, rRNA are known as non-coding RNA because it is coding for the proteins. And tRNA, rRNA, and other different RNAs, they do not code for any protein, and therefore they are known as non-coding RNA. So this is the importance of mRNA. Okay, chalo. One thing we know, that whatever amino acids has to be added in the protein, that is decided by mRNA, and also the sequence is decided by the mRNA. Okay. Now we know that XYZ amino acid has to be added. Now someone has to bring that amino acid to the ribosome, because ribosome is actually the place where synthesis takes place. For example, when we cook, okay, so when we cook, we have a stove and we have a utensil. We have a kadhai or a utensil, anything we have. We have a cooker, pressure cooker, let's say. So in pressure cooker, if we are making a lentil, if we are making any rice or a dal, so we put all the ingredients in the pressure cooker, and in the pressure cooker, in the stove, your actual cooking takes place. But for that actual cooking to take place, you have to open your jar, wash your rice or whatever, and you have to put that in that pressure cooker which is on the stove, so that your rice can be cooked. So you are doing that part of putting all the ingredients inside the pressure cooker. The ingredients are already made. If I say that I am making rice, I'm not going to the farm and I'm cultivating the rice. No, I have rice at my home. I'm just washing it and putting on my pressure cooker. Same is the function of tRNA. That ribosome is like a pressure cooker where proteins has to be made, and tRNA is like us who are washing the rice and putting it in the pressure cooker. So tRNA molecules are nothing but they are bringing the amino acid, just like we are bringing rice from a jar in our kitchen, and we are putting it in a pressure cooker in the ribosome. So that is the function of tRNA. tRNA brings the amino acid to the um, ribosome, so that peptide bond formation can be done by the ribosome. Okay, so mRNA is coding for the amino acids, tRNA is bringing the amino acids, and after the amino acid has been brought, the peptide bond formation, that is done with the help of rRNA. So rRNA is helping in the ultimately the peptide bond formation. So what is the function of mRNA, tRNA, rRNA in the case of translation? Always remember, first you need to you need to know the sequence, and that sequence is known by mRNA. After knowing the sequence, you should bring the amino acid that is brought by tRNA. After bringing the tRNA amino acids, the peptide bond formation has to be done, which is done with the help of rRNA. So 1 2 3, this is the sequential order of the of the role of RNAs, which are RNA, which is required in translation. Okay, so just we can read it over here, that protein translation by ribosome requires 3 types of RNA. Okay, mRNA, which specifies the amino acid sequence, ठीक है, tRNA, which converts the information in mRNA codons into amino acid. tRNA carry the amino acid sequence, and rRNA, which makes the bulk mass of the ribosome, that catalyzes the reaction in which the peptide bond formation occurs. Okay, so coding of the sequence, specifying of the amino acid sequence, mRNA, bringing the amino acid to the ribosome, tRNA, and formation of peptide bond, rRNA. Okay, so is this everything? Is this up till now? Is it clear to everybody? Is this thing clear to everybody? Okay, fine. Now we'll move next. So now we know these are the components which are required for translation and the role of different kinds of RNA in translation. Now we come to the structure of tRNA. Structure of mRNA I'm not going to deal, because that we have already dealt when you're doing transcription and all. Here, so there is some problem. See, yeah, I don't know what problem it is showing. Anyways, so yeah, so structure of tRNA. Now tRNA can be studied in 2 ways. Okay, one is the secondary structure and other one is the tertiary structure. So this is your secondary structure and this is your tertiary structure. So secondary structure is in the form of a clover leaf. This is a clover leaf structure. Okay. And tertiary structure is an L-shaped structure. So these points that I have written, they are important points. So that's why I wrote them here. Then if you look here, you can see the comparison also. This is the anticodon loop. This is the D loop. This is the T-cyc loop. This is the variable loop. And this is the acceptor arm. So amino acids comes at the acceptor arm. And acceptor arm is always at the 3 prime. I'll just explain you in this graphs. See, I don't know what is. Yeah. See. Look here, everybody. This is the acceptor stem. This green one is the acceptor stem. And even in this green one, this CCA is the region where amino acid comes and binds. This is the region where amino acid binds. Okay. Here the amino acid binds. So amino acid always bind at the acceptor arm at the 3' end, which ends with CCA. The last 3 nucleotides of this at the 3' end of the acceptor arm is CCA. Then this is the anticodon loop and this is the codon. 1, 2, 3, then it would be 3, 2, 1. Okay, 1 is always at the 5' end. So this is the 5' end, there it is 1. You can see this is the 5' end, so it would be 1. This is the 3' end. 3 ke side mein 3 hoga. So 3' end, so 3 would be there. Third codon is near the 3' end. Aise, you can remember it like this, that third codon is near the 3' end and first codon is then obviously would be at the 5'. Okay, and this is L-shaped structure. You can see like this inverted L. It's an L-shaped structure. So this is your secondary structure and this is your tertiary structure of tRNA. So mostly when if anybody ever asked you, mostly we draw the secondary structure because in secondary structure it is more clear. Fine. Now we'll try to solve 2 questions quickly. So which of the following RNA molecules serve as a adapter during protein synthesis? mRNA, tRNA, rRNA, and none of the above. I'll give you 1 minute to think and then I'll switch the screen. Okay, I'm switching the screen now. Let's see what you have answered. So Kavita has answered B. Janera has also answered B. Okay, others? Others also try to answer the question. Nobody wants to give a try? Okay. The correct answer is B. Because we have already studied adapter means which is adapter molecule means something that acts like a bridge. Okay. So between your amino acids and between your ribosome, these are your amino acids and this is your ribosome. The bridge is your tRNA molecule. This tRNA is bringing the amino acid to the ribosomes. So tRNA is like a bridge. It forms tRNA forms a bridge between amino acid and ribosomes and it acts like an adapter molecule. So the correct answer is B, that is tRNA. Okay, next question is the amino acid joins at the dash of the tRNA. D loop, variable arm, anticodon arm, or acceptor arm? Read the question carefully and then try to answer. I'll give you 1 minute and then I'll switch this. Okay. Okay, so I'll switch the screen now. Okay. Janara, Tarkeshwari, Kavita, everybody has answered D. And D is the correct answer. So we have already studied here. Yeah. So here you can see this is the acceptor arm. And in the acceptor arm at the 3' end, amino acid comes and bind. And 3' end with CCA. Okay. So now we'll see the genetic code. So whenever we study about genetic code, there are certain properties of genetic code that are very important to learn and remember. First is degeneracy of genetic codon. Second is genetic code is non-overlapping. And third is genetic code is comma-less. So there are many properties. I have written all the properties here. But degeneracy, non-overlapping, and this comma-less, these are very important properties. So first of all, what is a genetic code? So it is a set of 3-letter combination of nucleotides, which is called as codon, each of which corresponds to a specific amino acid or a stop signal. That means if this is your mRNA and this is ATT, AUU, GCUCCAAU. Suppose this is the sequence of your mRNA, let's say. Okay, so this mRNA sequence is clustered in the pair of 3. 1, 2, 3, 1, 2, 3, 1, 2, 3. And suppose if there is C also here. So 1, 2, 3. And this 3 nucleotides pair of, so not pair, but this combination of 3 nucleotide is your codon. This is one codon. So this is codon number 1, this is codon number 2, this is codon number 3, and this is codon number 4. And each codon specifies for one amino acid. Okay, so if I make it here for your, I'm just drawing it here so that it becomes even easier for you to understand. This is your mRNA 5' end to 3' end. You have A, U, G, 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 C, C, T, G, C, T, sorry, U, U, A, U, G, C, C, U, A, U, C, U, C, G. Whatever it is. Okay. So, now this is the sequence of your mRNA. Okay. This is your mRNA. So, this mRNA is divided into 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Okay. And suppose any other nucleotide. This is, so each, this is known as codon. So combination of 3 is known as codon. Now important point to, yeah, okay. important point to understand over here is that I am making this 3 frames. I can start from the first nucleotide, or I can make it like this: 1 2 3, 1 2 3, 1 2 3, 1 2 3, 1 2 3, 1 2 3, and 1 2 3. I can make it like this, and I can also make it in another way when I start from this: 1 2 3, 1 2 3, 1 2 3, and so on. So that means your this combination, this can be made in 3 ways. Okay. Now the question comes, which combination has to be selected? So by default, it selects the combination which gives it the start codon at the earliest. So now, according to this, can you tell me, if I name this particular combination as A, this particular combination as B, and this particular combination as C? So now can you tell me which combination should I choose, A, B, or C, depending on in which combination I am getting the start codon at the earliest? Tell me,
A.
A or B? A.
A.
A. Okay. Why? Because in A, at the first place only, I am getting the start codon. And if suppose if I choose C, you can see here also is AUG. This is also AUG. But 1 2 3, at the fourth place I am getting the start codon, but here at the first place itself I am getting the start codon. So your frames can be made in 3 ways. There are 3 combinations or 3 ways in which your frames can be made, but which way the frame would be made depends upon in which frame we are getting the start codon at the earliest. Okay. Now, this combination of 3 nucleotide is known as codon. So, this is codon 1, this is codon 2, codon 3, codon 4, codon 5, codon 6, codon 7, and so on. And each codon codes for one amino acid. Okay. Now, why I am telling you this is because on the basis of this only we are going to study our genetic code. How? First property of genetic code is that they are mostly triplet coded. Okay. You can see here they are triplet coded: 1 2 3, 1 2 3, 1 2 3. Fine. So this is done. Second property of genetic code is that they are universal in nature. So this is also, we know that whatever genetic code, it is same across prokaryotes and animals. So in prokaryotes and animals it is same, and throughout the animals it is same. Means ऐसा नहीं है कि in one animal AUG would code for something else and in another animal it would code for something else. Okay, there are some exceptions, but mostly the genetic code is universal. Okay, then they have a degenerate code. Now I'll just put this on hold. I'll tell you what is the meaning of degeneracy at the end. They contain start and stop codons. Okay, this is a start codon, and here somewhere, if it is say after this, if there is suppose this is a stop codon. So at the 5 prime end you will find a start codon mostly, and the 3 prime end of the protein will determine wherever the stop codon is there. Okay, this property is also fine. Then they showcase polarity. What is the meaning of polarity? That it starts from 5 prime end and ends at the 3 prime end. So this is your polarity. Here the polarity is 5 prime to 3 prime. 5 prime to 3 prime describes polarity over here. Fine. Then the code is mostly non-overlapping. What is the meaning of this? Non-overlapping means that, see here, if I am taking this A frame, AUG, GGC, CUG, this AUG is individual, CG is individual identity, means it's a separate identity. This G is only present in this codon. This G is not coming here also. The code is not like this. AUG, GGG. Suppose if I have AUG and GGC. Okay. This is one codon. This is another codon. These two codons are separate and there is no overlapping between the two codons. Now, what is the meaning of overlapping over here? That when I make codons, I do not make like this. AUG. First codon, I write AUG. And then if I am writing GGG, second codon as GGG, then this G is overlapping in first codon also and second codon also. Okay, but this is not the case. We do not do like this. We keep each and every nucleotide separate. If G is present in first codon, it will not be present again in second codon. In second codon, another G is there, but not this G would come. Okay, so it is not overlapping. And then they are comma-less. That means when we are writing codons, uh, yeah, a combination A, you start codon, yeah, fine, Madhvi, you're saying it right. Sorry. Huh. So they are comma-less means whenever you are writing the codons, you're not writing any comma over here. Like when I write the sequence here, have I kept comma anywhere? No, they are comma-less. Okay. Now comes this point that we have left, which is degeneracy. Now this point is very important. Why? I'll tell you. Let's take an example. Okay. Okay, this, just a minute. Yeah, this was our last week's class. No, I just want to take this figure here to understand you something, to make you understand something. So if I talk about alanine, or let's say leucine, leucine is a better example. Okay, so I am talking about leucine here. Now try to understand this, because why I am putting more stress on the topics that I am teaching today, because what is translation, what are the steps, and what are the factors, what are the enzymes, what are the elongation factors, initiation factors, termination factors, these you will get in each and every kind of notes. There are so many articles, there are so many notes, there are so many reference books, pathfinder is there, everything is there, so you will get the names easily. But the concept building is important, because if your concepts are clear about translation, then you can remember the rest of the names. Remembering the names is not important here. Knowing the concept and understanding the knowledge of it is important. So this degeneracy, why I'm telling you that even in PhD, many of the students get confused what is the meaning of degeneracy of codon. Something that we are studying right from our bachelor's. So here, you see here correctly, leucine has two codons. Okay. It can be coded by U, C, sorry, where is leucine? U, U, A, UUA. You can see this is your leucine. UUA and UUG. UUG. So this means here also you can see tyrosine can be coded by two codons. Then cysteine can be coded by two codons. Leucine can be coded by 4 codons. Okay. Here you see threonine, it can be also coded by 4 codons. So that means one amino acid can be coded by more than 1 codons. But one codon cannot code for more than one amino acid. What does it mean? That leucine can be coded by UA and UUG. But this individual UUA can only code for leucine. It cannot code for any other amino acid. This property is known as degeneracy of codons. What is the property? That one amino acid can be coded by more than one codons. This is known as degeneracy of codons. That means codons, they are degenerate. That means codons can be coded by more than one amino acid. Is this clear to everybody? What I'm trying to tell, is it clear or not? Okay. I hope it is clear to everybody. Good. Fine. That means one amino acid can be coded by one codon. This is the degeneracy of codon. But always remember that one codon cannot code for more than one amino acid. That means leucine can be coded by UUA and UUG. But UUA will always code for leucine. It will not code for any other amino acid. And this is very important in genetic code to understand. So this is what degeneracy of codon means. The coding is degenerate, which implies that more than one base triplet codes for the same amino acids. This is a writing language, but I told you in a much simpler way. Okay. Now this codon anticodon base pairing. Now this codon anticodon base pairing I have already told you in the last class a very great detail. So I'm not going to repeat that. I don't know why there is some problem here, why it's coming every time. Fine. Just you need to understand that there are three nucleotides that makes a codon. One, two, three. One always, first nucleotide is always present at the 5' end. Third nucleotide, which is the last nucleotide, it is always present at the 3' end. And third nucleotide is, this position is known as a wobble position or a wobble base, because here also you can see, if you could see here, UUA, UUG. So third position is a wobble place, means if one amino acid has more than one codons, it is actually at the third position where the difference comes. You see, UU is same here, the difference is at the third position, so, which is known as a wobble base. So one thing which we got to know is first base will come at the 5' end, third would come at the 3' end. So codon anticodon base pairing means this is the tRNA and this is the anticodon. Okay, here you have 3' end. If here you have 3' end, then it would be 1, 2, 3. This is the 5' end. This is your tRNA. I'm just making the anticodon loop. Fine. Why? Because first nucleotide is near the 5' end, third is near the 3' end. Then how the codon would be arranged? This is your anticodon. Then how would be the arrangement of codon? This is the mRNA. This would be 3, 2, 1, and this is your 3' end and this is your 5' end. And this is your codon. Is it clear to everybody? 1 is pairing with 3, 2 is pairing with 2, and 3 is pairing with 1. This is how codon anticodon base pairing occurs. Now comes to the basic okay, before coming to charging, first we'll try to solve this question and then we'll come to okay, we have to read about charging. Fine. Now we come to charging of the tRNA molecule. Now what is the meaning of charging of tRNA? Last time also. So yeah. So if this is your mRNA, this is your ribosome smaller subunit, and this is the larger subunit of ribosome, And this is your tRNA molecule. And this is your amino acid. Something like this is the structure. Fine. So, attachment of the amino acid to the tRNA, this attachment is known as charging. So, if I say a tRNA is charged, that means amino acid is attached to the tRNA. And this charged tRNA only comes to the ribosome. So, always remember it is not like that. That first tRNA will come on the ribosome and then amino acid will come. No. tRNA is an adapter molecule. So it is bringing the amino acid. So tRNA plus amino acid, it will come to the ribosome. Okay? Always remember this. So this attachment of amino acid to the tRNA molecule, this attachment is known as charging. Sorry. Charging of the tRNA. Now let us look at how it happens. So charging of tRNA is a two-step process which involves first is activation of amino acid and second is attachment of the activated amino acid to the tRNA. That means first amino acid, see amino acid has to attach to tRNA molecule. But now as I said that the attachment of amino acid to the tRNA molecule is a energetically expensive process. That means amino acid cannot attach to the tRNA molecule by its own. It needs some energy. So amino acid gets this energy, that energy in the form of ATP. So first what happens, that amino acid, it attaches to the ATP. And then this amino acid becomes amino acyl AMP. You see? Then this amino acyl AMP comes to the tRNA molecule. And then this whole tRNA becomes amino acyl tRNA. Okay. And in this process, the enzyme which catalyzes this reaction here and here, it is amino acyl tRNA synthetase. Is it clear? So what is happening? First, amino acid attaches to ATP and amino acid gets the energy. So it becomes amino acyl AMP. That means from ATP it has taken just one phosphate. These two phosphates are removed. It becomes amino acyl AMP. Then what happens? This amino acyl AMP then comes and binds to the tRNA molecule and AMP is released. So this is the tRNA molecule. You can see this amino acid then binds to the tRNA and it becomes amino acyl tRNA. And this whole reaction is catalyzed by amino acyl tRNA synthesis. Is this clear to everybody? Charging of tRNA, is it clear? Yes or no? Okay. So now we'll try to solve two questions and then we'll move to the mechanism of translation. Okay, so slowly we are studying today because we are focusing only on translation, because last class we could not study it in that much detail. So I promised that I will explain this translation again. So that's why I'm doing that. Okay. So the question is, identify the correct statement. Now this is a tricky one. One amino acid is coded by only one codon. Okay. Many amino acids can be coded by one codon. Okay. Many codon can code for one amino acid. Many codons can code for same amino acid. These two options would be same. So I'm making it many. Many codons can code for many amino acids and many codons can code for same amino acid. Now tell me which is the right answer. I'm giving you 2 minutes for this. Try to read each and every statement and then think what we have learned right now and then answer the question. 2 minutes for this question. Okay, just a minute. I'm seeing whether you are attempting or not. Okay, so I think three. Madhvi has answered it as C, Jahanara as C, Kavita as D. Okay, others also please try to answer the question. Others, anybody else who wants to give it a try? No? Okay. Let's try to understand each and every statement one by one. One amino acid is coded only by one codon? No, because we have just saw that here in this example that leucine is coded by UUA and UUG. Leucine is coded by UUA and UUG. That means one amino acid can be coded by more than one codon. So this is a wrong statement. Many amino acids can be coded by one codon? No, because we studied that one codon can only code for one amino acid. But one amino acid can be coded by more than one codon. This is what we have studied. This is what all degeneracy is about, right? Then many codons can code for many amino acids. This is a very vague statement. Why it is a vague statement? Because there is nothing is specified over here. Many codons can code for many amino acids. Suppose if those many codons are UUA and UUG only, then are they coding for many amino acids? No, they are coding for same amino acids. So that means this statement is not a complete statement, because many codons can code for many amino acids makes no sense. You have to be a little more specific. Now, what is a D statement? Many codons can code for same amino acids. Now, this makes sense because here, if I'm taking one example, so all the statements I am explaining you with the help of this example only. So, you can see that many codons they can code, many codons can code for same amino acid. So, this makes sense. The correct answer is D. Is this clear to you? Is this question clear to everybody? If no, then please ask me your query or problem. So Kavita has answered it correctly, and I hope that Madhavi and Jahanara, the whatever problem that you had or confusion you had, it is solved now. Fine. Next question is, identify the correct meaning of amino acyl tRNA. Amino acyl tRNA. What is the meaning of amino acyl tRNA? tRNA is charged with ATP. tRNA is charged with an amino acid that corresponds to anticodon of mRNA. tRNA is charged with GTP plus amino acid. tRNA is charged with amino acid that corresponds to its anticodon. I'm giving you 2 minutes. Read all the statements slowly and then try to answer. So are you getting the question? Okay. Okay, I'm switching the screen now. Let us see how many of you have answered it correctly. Okay. So Janara has answered A, Madhvi has answered D, and Kavita has also answered D. Let's see. Okay. So amino acyl tRNA. So tRNA is charged with ATP. tRNA is charged with an amino acid that corresponds to anticodon of mRNA. tRNA is charged with GTP plus amino acid. tRNA is charged with an amino acid that corresponds to its anticodon. So charging of tRNA means it does not mean that tRNA has ATP. No. Charging means if I say tRNA is charged, that means tRNA has its cognate amino acid on it. So when amino acid comes and joins tRNA, then that is known as charging. In that process, ATP is required. That is a different thing. This is an energetically expensive process. So in this process, ATP is required. That is different. But the charging of tRNA means that amino acid has come and bound to the tRNA. Okay. So the correct answer would be D. Fine. Now we'll come to the mechanism of translation. So up till now we have studied all the prerequisites. Prerequisites means what are the things, what are the properties of translation, what are the different kind of molecules. Most of the molecules, not we have covered all the transcription factors and sorry, translation factors and all, but we have tried to cover the basics that are required for a translation process to happen. Now we are going to see the mechanism of translation. So mechanism of translation means that how translation actually takes place. So translation takes place, there are three steps: initiation, elongation and termination. Now we'll come to the initiation. Now always remember, when we talk about prokaryotic translation and eukaryotic translation, initiation is the main process which has most of the differences. The logic remains the same, but differences are there. And initiation is the one which has a lot of steps also that you need to understand and remember. So, see, I'm just doing like this. Yeah. Now you see here. So, this is the small ribosome unit. So, one thing always remember, when we talk about initiation, so nothing is there. It has to start from the scratch. When I say it has to start from the scratch means even the two ribosomes, if you're talking about eukaryotic, a 40S ribosome and a 60S ribosome, even these two ribosomes are not together. So when the transcription is started, these two ribosomes are separate. So you can see this is the small ribosome unit. So what happens to initiate the transcription? This is the charged tRNA. Okay, this charged tRNA, which is the tRNA which contains the amino acid, that is fine. Now the first tRNA always, or in most of the cases, it contains methionine, AUG. Okay, AUG, and the anticodon would be UAC. So first tRNA which has methionine in it, that tRNA is known as the initiator tRNA. Why? Because this tRNA is going to initiate the translation reaction. That is why this tRNA is known as an initiator tRNA. Now, this initiator tRNA will come, it will bind to the small ribosomal unit. Okay. Now, this initiator tRNA and small ribosome, now it will come and it will go and bind to the mRNA. So always remember how it is like this. First, tRNA comes. First, tRNA comes and it binds to the 40S ribosome. Then this complex binds to the mRNA. And then in this complex, large ribosomal unit, that is 60S, comes and binds. Okay, so you can see this is the initiator tRNA and this is the ribosome. So first initiator tRNA will come, it will bind to the ribosome. Then this complex will bind to the mRNA. Now, how this complex will bind to the mRNA? So this complex will scan the mRNA to find where is the start codon. Because this complex has to bind at the start codon, near the start codon only. Because UAC has to bind with AUG, na? This anticodon has to bind with the codon. So this whole complex will come right from the 5' end. 5' there is a gap. It starts scanning. It scans where is the stop codon. See, complex scans to find the start codon. Now this complex will scan. Okay, where it is, where it is, where it is. Okay, acha, there is your start codon. Now, once the complex finds the start codon, it will stop there. And now, since the anticodon and codon has paired, then your large ribosomal subunit will come and bind. And then it will form the initiation complex. Okay. So when I say initiation complex, so initiation complex would have mRNA plus 40S ribosome plus 68 ribosome plus initiator tRNA. See, this is your mRNA. This is your smaller subunit 40S ribosome. This is 60S ribosome and this is your initiation complex. So when all the things are combined together, then it forms the initiation complex. And this is what translation initiation is about. Translation initiation means formation of the initiation complex, how initiation complex forms. So this is how initiation complex forms. This I have explained you in a very simple way. Simple means in a less descriptive way. Now we'll try to understand this whole that we have studied in a little bit more detail. But is this diagram clear to everybody? Is it clear to everybody? Yes or no? If you have doubts, you can ask me. Okay. Okay, fine. Okay, so now we'll try to understand this particular process in a little bit more detail. Just a minute. Okay. Now, try to follow this diagram and the points, and everything would be very clear to you. So, this is your initiator tRNA. Okay. First, I'll explain you the diagram and then I will explain you how is it happening. This is the initiator tRNA. This initiator tRNA First, one transcription factor, which is eukaryotic initiation factor 1, initiation factor 2. This initiation factor is already bound to a GTP molecule. This initiation factor will come and it will bind to the tRNA molecule and it forms a ternary complex. Okay, this is the first step, formation of a ternary complex. Now, why it is known as a ternary complex? Because it has three components in it. And what are those three components? Initiator tRNA, which has methionine in it, GTP molecule, and this eukaryotic initiation factor 2. There are three components, therefore it is known as an ternary complex. Now this ternary complex will bind to the ribosome. That is what we have studied over here. That first this binds and then it binds to the ribosome. So now why this GTP and eukaryotic initiation factor? So this eukaryotic initiation factor helps in taking this charged tRNA to the ribosome, and this requires energy, so it is bound to GTP. So now you see this is your 40S ribosome which has mRNA in it. Then what will happen? Don't consider this mRNA that it is getting bound to the mRNA itself. This mRNA is there, but first it is not going to bind with the mRNA completely. It will just be there. The perfect binding will happen later on. I'll explain you. So what is happening here? This ternary complex is formed. Okay. This is the initiator tRNA. This is the transcription, this is the initiation factor along with GTP. 1, 2, and 3. 1, 2, and 3 ternary complexes form. Now this ternary complex will bind to the 40S mRNA and it will become 43S. Why it will become 43S? Because 40 plus 3 ternary complex, 3 components are binding, it becomes 43S. Now in this 43S complex, 60S subunit, that means the larger subunit will come and bind, and now it will become 80S. Now this initiation complex is bind. Now it will scan the mRNA, it will find the start codon and it will attach there. Here it is only present, but scanning happens only after this 80S complex is formed. The whole initiation complex is an 80S complex. Now this 40S and 60S, the two subunits are separated together. So obviously there are certain initiation factors that are bound that keeps them in separated mode. And those initiation factors are EIF3 and EIFA. So the 40S ribosome is bound by a eukaryotic initiation factor EIF3E and EIF1A, which prevent the 60S subunit reassociation with 40S. Okay, fine. Next is the first tRNA that comes is the methionine tRNA, initiator tRNA. Okay, the methionine tRNA gets bound by EIF2 and GTP and forms a ternary complex. Okay, this ternary complex is formed. Now what happens? This ternary complex binds to the 40S ribosome. This is the 40S ribosome and gets converted into 43S pre-initiation complex. So this is 40 plus 3. It is a 43S pre-initiation complex. It is not a complete initiation complex. It's just a pre-initiation complex. Then what happens? Now this formed 43S pre-initiation complex binds to mRNA and forms 48S initiation complex. Okay, now what happens? Once the tRNA binds to the ribosome and then it goes and it binds to the mRNA. Here what we have studied. See, tRNA first bind to the ribosome and then this whole goes and binds to the mRNA. That is what it is written. That from 43S it becomes 48S. 48S initiation complex means initiation complex where ribosome plus tRNA binds to the mRNA. Now this 48S complex scans the mRNA to find the start codon. and attaches there, and then the 80s ribosome comes and binds the 48S complex. And then this is how the transcription initiation is completed. That means all the machinery that has to happen, it is formed. Just read these points by yourself. I'm giving you 1 minute. Read these points, and if you have any doubt, ask me. This is important because I'm telling, because this is important. And then if you have any doubts, ask me. In 1 minute, try to read the points. Try to read each and every point very carefully, and then if you have a problem, ask me. Okay. I hope you have read the points. So, ma'am, repeat the first 3 points where EIF factors are involved. First 3 points. Okay. So, see, what has to happen is that this is the tRNA which has methionine. This tRNA which has methionine, it has to bind to the smaller subunit of ribosome. This is 40S. But this, even though this tRNA is charged, means it has methionine, this cannot bind to the 40S ribosome on its own. Why? Because binding to this 40S ribosome is again an energetically expensive process. So first, addition of this methionine is also requires energy, and binding of this whole charged tRNA to the smaller ribosomal subunit also requires energy. Okay, and some factors. So now this charged methionine knows that it has to go to 40S ribosome, but it does not know how to do it. So just like we have Google Maps, so Google Maps gives us direction where to go. Similar to Google Map is the work of eukaryotic initiation factor 2. Eukaryotic initiation factor 2. So this comes as a savior, and it tells to this charged tRNA that don't worry, I am here to help you. Take me along with you, and I will guide you to the smaller ribosomal subunit. But then there is a problem. What is the problem? Just like Google Maps needs what? Internet connection. Internet connection is the energy that Google Map gets, and then only it can gives us direction. Similar way, eukaryotic initiation factor 2 also needs some kind of energy so that it can do its work, and that energy it gets from GTP. So this make it as a story, a cartoon story, that this is the initiator tRNA. It is saying that please help me, I have to bind to the smaller ribosomal subunit. This eukaryotic initiation factor says, okay, I will help you. It charges itself by attaching to the GTP, and then this whole complex comes and binds to the initiator tRNA. So eukaryotic initiation factor 2 plus GTP plus this initiator tRNA. There are three components, 1, 2, 3. It becomes a ternary complex. So now, with the help of eukaryotic initiation factor 2, EIF2 and GTP, this initiator tRNA binds to the smaller subunit. Okay. So smaller subunit is 40S. Now in this 40S subunit, three more components, 1, 2 and 3. Three more components are getting attached. So it gets converted to 43S. So now this 40S subunit becomes 43S. Now in this 43S subunit, what happens? This 43S subunit, which has ribosome, tRNA and eukaryotic initiation factor 2 GTP. Now this whole goes, and this is your mRNA. It scans the mRNA to find the start codon. Once it finds the start codon, it fixes here, and then the larger subunit comes, and then it forms the initiation complex, whole initiation complex. So the whole initiation complex only becomes the complete initiation complex is formed only when both large and small ribosomal subunit are there, and they are attached at the specific site in the mRNA, which is at the start codon. Is it clear? Anybody has any confusion, please ask me.
What about 48, ma'am?
48. See, if you see in this diagram, sorry, here, 43S pre-initiation complex binds to the mRNA and form the 48S complex. That means what I'm trying to tell here, up till 43S it is clear now. This is your mRNA. This is the smaller ribosome subunit. This is the initiator tRNA. This is eukaryotic elongation factor 2 and GTP. This is the start codon. Now, this whole 43S plus mRNA, it becomes 48S. Okay, that means 43S, which has ribosome, tRNA, EIGF2 GTP plus mRNA, it becomes 48S. And in 48S, when 80S subunit comes, it becomes initiation complex. Is it clear?
Yes, ma'am.
Okay, fine. So why I was giving more time on this is because it's very difficult to find an explanation of how the different complexes are formed in eukaryotic initiation. It becomes very confusing. Like even if you are reading textual matter, textual information in books, it becomes difficult for you to, it becomes, because it was difficult for me also to understand that how this initiation complex is forming, what is ternary complex, what is 40, what is 43, what is 48, what is it. So this is a little difficult, but if you are studying like this, like the way I told you, It is very easy for you to understand. So these slides, I would request everybody to download it, because not just for your this thing, for this I mean this course per se, but in general it would be helpful for you. Although it does not has complete information, because I have not included the information that needs to be learned by, like, that needs to be that I mean that you have to learn. I have not included those information. I have only included information that you need to understand, where some logic is involved. Things that you just need to rectify, that I have not included. But if you know these much points also, that thing, that part you can find in any book. That is not a difficult point. Okay. So this is about translation initiation. Now what is there? Up till here it is clear. So now you have mRNA, you have a smaller ribosomal subunit, large subunit, and the large subunit has like three sites: EPA, EPA, and the first tRNA, that is the initiator tRNA, it comes to the P site. So this is the initiator tRNA, which is present at the P site. You can see over here in this diagram. See here, smaller subunit, larger ribosomal subunit. This is the mRNA, and first tRNA, which is the initiator tRNA, which is at the P site. Now, what is the meaning of elongation? So, elongation involves three steps primarily. First is binding of the next tRNA to the A site. So, always remember the first tRNA, which is the initiator tRNA, that comes at the P site. First tRNA. Ma'am, first point, didn't get it. EIF3 and EIFA. Okay, just a minute. Okay. So basically, you always remember this is the smaller ribosomal subunit and this is the larger ribosomal subunit. Before the start of transcription, these smaller and larger ribosomal subunits are separate. They're not together. So, in order to keep the two subunits separate, some kinds of initiation factors, which are known as eukaryotic initiation factor 3 and eukaryotic initiation factor 1A, they are bind to the smaller ribosomal subunit. If this is your smaller ribosomal subunit, and these are the factors EIF3 and EIF1A, they are bound to the smaller ribosomal subunit. Why? So that larger ribosomal subunit cannot come and bind to it. These factors prevents the binding of the larger subunit. So when here at this point, when larger subunit has to actually bind, at that time these factors are removed. Okay. So if you know poly A, you know about poly A binding protein? Anybody? Poly A binding protein. You know the function of poly A binding proteins? Anybody has heard of its function? So basically, poly A binding proteins are the proteins that binds to the denatured DNA at the time of replication. So it maintains them in single state so that they cannot come and again form a double-stranded DNA. So just like to keep the two ribosomal subunit separate from each other, the smaller ribosomal subunit is bound by some factors, eukaryotic initiation factor 3 and eukaryotic initiation factor 1A, which prevents the premature binding of larger subunit to the ribosome. Only when the 48S complex is formed, Only after the formation of 48X complex, the 80S, the larger ribosomal subunit, has to come and bind to the smaller subunit. So at that time they are removed. EIF3 and EIF1A are removed so that it can come and bind. So you can imagine that when these proteins are bound to the smaller subunit, they are masking the attachment sites of the larger and the smaller subunit. And that is why the larger and the smaller subunits are unable to bind to each other. Is it clear now, Madhvi? Okay. Yes. Yes, I am talking about yes, I'm talking about single stranded binding protein. So single stranded binding proteins has a work that it prevents the DNA from again getting double stranded. Okay. Similarly, just like the function of single stranded binding protein, the function is eukaryotic initiation factor 3 and EIF1A has similar function just like that of SSBP, single stranded binding protein. Okay. Okay. Now we'll come to translation elongation. So, translation elongation involves steps. First step is binding of amino acyl tRNA to the A site, peptide bond formation, and translocation. So, what does it mean? That the first tRNA, whatever is the first tRNA, which is the initiator tRNA, it always comes to the P site, and subsequent tRNAs, it comes to the A site. And then from P to A, there is a peptide bond formation. And then the tRNAs translocate from A to P to E. This is your elongation. So let's try to understand this elongation in a little bit detail. See, what I can do is that I can reduce uh, I'm trying to reduce these size so that now you see here. Not laser. I want pen. Hmm. The P site of the ATS complex has methionine tRNA. See here, Abhi, not just in this diagram, I'm just telling you an A site is free. The amino acyl tRNA will be placed at the A site. What does it mean? That the first tRNA, that is the initiator tRNA, which has methionine at it, it comes to the P site. So the next tRNA with the next amino acid, it comes to the A site. Okay. Then what happens? The peptide bond formation is catalyzed by the enzyme peptidyl transferase. This enzyme performs its activity on 28S RNA of the 60S subunit of the ribosome. Okay. Hence, peptide bond formation occurs by the attachment of the growing peptide chain with tRNA at the A site. So, what happens? You see, this is the amino acid and this is the methionine. So, there is a peptide bond formation that happens here. So you can see this is methionine. It comes here and the next amino acid is here. So peptide bond formation is happening at A position. So what happens? This methionine will go here like this and it will form a peptide bond. Then what will happen? The ribosome will then move to the 3' end of the next codon of messenger. Then what will happen? This will move here, it comes here, it does not has any tRNA now, and this moves here. Then next tRNA will come, and then again the process is repeated. Mm. Okay. Is it clear to everybody? See here. This is P, where you have the first tRNA. Next tRNA comes to the A site. Okay, two tRNAs are there. And then formation of peptide bond happens. And this methionine will go to the A site. So methionine is at the above. And next is your next amino acid. Now this has no tRNA. So it will go here. And then from here it will exit. And this, it will go here. It comes here to the P site. So if this is your E, P and A, P has methionine and A has some other amino acid. I am labeling as it amino acid 2, and this is methionine. So what happens? This methionine will go to the A site, and then ultimately this P, it has no tRNA now. At A position, you have amino acid 2, and on the top of this, you have methionine. This is EPA. Now this which has no tRNA, it will go here, and from here it will go out, and this will come here. And then again A would be free, and then in A, next amino acid will come. Okay? Is this clear or not? Is it clear to everybody? How formation of peptide bond is taking place and how it is moving? Okay, fine. So always remember, one thing that we have to remember is that first tRNA, which is the initiator tRNA, it comes to the P site. The next amino acid tRNA, it comes to the A site. And it moves like from A to P to P to E. E is the exit site. Okay. Now we come to the translation termination. So there are two stages of termination. First is cleavage of the growing chain, and second is your recycling. Now what is the meaning of cleavage? One of the stop codon UAA, UAG or UGA will terminate the growing polypeptide. Okay. In eukaryotes, all of the three stop codons are recognized by one eukaryotic release factor, that is RF1. And eukaryotic release factor 3 helps to stimulate transcription, termination. Okay. So eukaryotic release factor 1, which recognize all three stop codons in the case of eukaryotes. Okay, fine. Then what is the next point? When the ribosome comes in contact with the stop codon, there will be no tRNA available for binding to the A site, and instead of it, a release factor will bind to it. After the binding of the release factor, the ribosome will fall apart into the following components. Small and large subunits would be released, peptidal product would be freed, and tRNA would also be free. So what happens? See here. So here, instead of any amino acid, stop codon comes. So when stop codon comes, then there is no tRNA that is specific for stop codon. So no tRNA comes, then release factor comes here. When there is a stop codon, then release factor, which is release factor 1. So release factor is inserted into the A site. And as soon as release factor is inserted, then this complex starts disassembling. What is the meaning of disassembling of the complex? That see, this tRNA is released, polypeptide is free, small ribosomal subunit, larger ribosomal subunit and smaller ribosomal subunits, they also go away. So that means as soon as the the ribosome recognizes a stop codon, then there is no tRNA that is specific or against the stop codon. In this case, release factor comes and binds to the A site. As soon as release factor comes and binds to the A site, the whole complex gets disassembled and it is translation is terminated. Is it clear? Is it clear
Yes,
to
ma'am.
everybody? Okay. So translation is not that difficult. Most of the problem, not problem, but the most complex portion of this translation is initiation only. If you are clear with initiation, then it's easier for you to understand. Fine. So, like we were studying, that there are um, two stages. First is cleavage of the growing chain. Okay, now we studied how the growing chain is getting cleaved. Second is recycling. You know what is recycling? Let us see. So, recycling of ribosomes always occur in eukaryotes. When the polypeptide and release factors are released, the ribosome are still bound to the mRNA and have the deacylated transfer RNA and P and E site. Okay. For the new polypeptide synthesis, these mRNA and tRNA must be separated and the ribosome must also be dissociated. All the process are collectively known as ribosome recycling. So ribosome recycling means, it just means that after your translation is completed, your ribosomes, your tRNA, your mRNA is not degraded, okay? They are still present. They will be used for another round of translation. So they are recycled. Just like we have a bucket, so when we go and take a bath, we fill the bucket with water. And after shower or after we take the bath, we don't just throw away a bucket, right? We use it the next day when we again taking the bath. Similarly, bucket is your ribosome. Ribosome is a utensil. Just like we have utensils in our kitchen, we have pressure cooker, we have all other utensils. So we are reusing it. We are washing it and we are reusing it. For one round of cooking we use, we wash it, we keep it. For another round of cooking, we again use it. Similarly, these ribosomes, these tRNAs, these are used one time for termination. They are not discarded. They are not ubiquitinated for death. They will be reused for next round of translation. Okay, so this is your recycling or reusing of all the components which are involved in translation. So I think we have completed the translation. Now quickly we'll try to see some of the questions.
Ma'am, one small query, ma'am.
Yes.
You said, as you said, 48, yes, it is combined with the, it is bind with 80s, is formed into 80s. It is in this 48 years, there
Yes.
are initiating factors are there. You, as you said the earlier, that is prevent the combined with 60 years, isn't it, ma'am?
No, no,
How could it?
40,
How
40.
could it? It is combined with after formation of the 48 years to the 80 years formation, ma'am.
Yes. So see, see here, if you just see, it's a very good question. Uh, yeah. Mm, where is the diagram? So 48S means that there is no 80, the 80S, that means the larger subunit has yet not added here. Okay. 48S initiation complex is formed as soon as the 43S complex binds to the mRNA. Now, after the 48 complex is formed, what next? 80S has to come, but 80S cannot come if eukaryotic initiation factor 3 and eukaryotic initiation factor 1A are born. So, first 48 S complex is formed. After then, simultaneously, like 80S big larger subunit is coming, and these factors are also removing. So as soon as like one factor is coming to the complex and one factor is getting removed from the complex after the formation of 48x, because these factors are inhibiting the binding of 80s to the 48s complex. So if 80s has to come and bind to the 48 complex, this has to be removed. So these are removed. It's like a simultaneous reaction, that okay, now there's a signal that 48x is formed. Okay, now these factors would be removed and 80s will come and bind to the complex. So 80s cannot come and bind to the 48x complex if eukaryotic initiation factor 3 and eukaryotic initiation factor 1a are present. They has to be removed before the actual joining of the 80s to the 48s complex. Am I able to answer your question? Otherwise, you can type it here because I am unable to listen you properly. Maybe because of network issue. Is that clear to you? Okay. You have written yes. Fine. Okay. So, just for your always remember, first there is ternary complex. Okay. And this ternary complex has methionine, methylated tRNA, and then eukaryotic initiation factor 2 and GTP. This is the ternary complex. Ternary complex binds to the 40S ribosome and it forms the 43S pre-initiation complex. This is known as pre-initiation complex. And this 40S is already bound with some initiation factor. Initiation factor EIF3 and EIF1A. Then this 48 43 is initiation complex. It scans and binds the mRNA and then it becomes the 48S complex. Now in this 48S complex, 80S comes and bind and it becomes the initiation complex. And when this 80S is binding, at that time, two factors are removed: eukaryotic initiation factor 3, eukaryotic initiation factor 1a, they are removed. And then this is bind. Is it clear?
Yes, ma'am.
Now, what I am telling you again and again, that I have explained you just 2 or 3 factors. But there are n number of factors. There are 7 to 8 more factors that are involved here. I have not included them because then it would be really a mess for you. It will become really confusing for you to understand. These are the factors which actually makes the translation initiation process. Other factors also have roles. You can just read it. That's not a very big thing, but these are the factors that really creates a difference in the translation initiation. That's why I have included these factors. But see, there is no limit to go in depth of any topic. Okay, you can keep on going in detail of each and every topic. But this is what, if anybody asks you about translation, then this much is, according to me, is enough. And then you have some diagrams that are given in CERT's lectures also. You can also go through them. Anyways, now we'll try to solve the questions. So the first question is, in protein synthesis, translocation is initiated with the movement of tRNA from P to the A site, dipeptidyl tRNA from A to P site, tRNA from A to P site, tRNA from P to E site. Think and answer the question. Hmm. Okay, I'm switching the screen. Let me see what you have answered. A and B. Janara has answered A. Kavita has answered B. Okay. The correct answer is B. Why? This is E, this is P, and this is A. E has methionine tRNA. A has some other tRNA with some other amino acid in it. What happens for the first? Here we are talking about initiation, the first bond and the first movement. This methionine will go and bind to this. So what will happen? This is E, this is P, this is A, this is empty. At the A position, we have amino acid and on above methionine. So we have this thing, two amino acid bind. So we have a dipeptide. And this then moves from A to P, and this empty moves from P to E. So translocation is initiated. This is the first translocation that takes place from the A side to the P side. Okay, this is a dipeptide. This is a di means two. There are two, and this moves from the A side to the P side. So that is why B answer is the correct one. Okay, next question is, this is incorrect about the nature of genetic code. Which of the following is incorrect? Genetic code are universal, overlapping, comma-less, and triplet. What is the incorrect one here? Which is the incorrect statement? Okay, I'm switching the screen. B. Yes. So B is the incorrect one, which is overlapping, because we have already studied that genetic code is a non-overlapping sequence. Okay. So B is the incorrect one. Fine. Next question is, in translation, this is not an essential component. Which is not an essential component? Amino acid, ligase, mRNA, anticodon. So which is not an essential component in translation? Tell me. Okay, I'm switching the screen. Okay. B. So Janara, Kavita, everybody has answered B, which is ligase. Yes. So ligase is not an essential component. Obviously, amino acids are required. mRNA has the code, anticodon, which is present in the tRNA, is required. But nowhere we have mentioned about ligase. So ligase is not an essential component. Then this identifies a particular amino acid and its cognate tRNA. Which identifies a particular amino acid? Topoisomerase, rRNA, ribosome, tRNA synthetase. Think. So, Madhvi, Movin, Jahanara, Movin, Kavita have answered D, which is tRNA. Yes, the correct answer is tRNA synthesis. Fine, you are able to understand now. It seems. Now, next question is, in which of the following directions does the polypeptide synthesis proceeds? From both N terminus to C terminus, either from N terminus to C terminus to N terminus, or N terminus to C terminus. Think and uh, find the correct answer. Okay, I'm switching the screen now. So, Kavita has answered D, Madhvi has answered D, and Janara has also answered D. N terminus to C terminus. Yes, it is the correct answer. So it cannot take place either from N terminus or C terminus. No, it cannot be from both. It has to be in just one direction, and that one direction is N terminus to C terminus, 5 prime to 3 prime. Now I have told, I think, the answer. Okay. The ribosome reads mRNA in which of the following direction? Both 3 prime to 5 prime and 5 prime to 3 prime. Either 3 prime to 5 prime or 5 prime to 3 prime. 3 prime to 5 prime direction only, or 5 prime to 3 prime direction. Tell me the correct answer. Okay. Madhvi has answered D. Momin has answered D. The correct answer is D. That is in the 5 prime to 3 prime direction. Okay. Next question is, in prokaryotes and eukaryotes, multiple ribosome can bind to a single mRNA transcript and give rise to beads on a string structure. What is this structure called? Monosome, multiribosome, oligosome, or polysome? 1 minute and then I'll switch the screen. Okay, let me see what you have answered. So, Kavita has answered D, Janara has answered D, and Madhvi has also answered D, and D is the polysome, and it is your correct answer. Fine. Next question is, with the help of which of the following proteins does the ribosome recognize the stop codon? Okay, cleavage factors, elimination factors, termination factors, or release factors? Think and answer. Okay, and I'm switching the screen. Let us see what you have answered. So, Kavita has answered D, Madhvi has answered D, and Jahanara has also answered D, which is the release factor, and this is the correct answer. So, with this, we have completed the translation. Just quickly revising what we have studied very quickly. So we have studied what are the components which are required for translation: mRNA, tRNA, ribosomes, various factors and enzymes. Then we studied about the role of different RNAs in translation: mRNA, rRNA and tRNA. Then we studied about the structure of RNA, the secondary structure and the tertiary structure, and the difference between the two structures. Then we studied about the genetic code and what is the meaning of degeneracy of codons and non-overlapping and commaless. Okay. Then we studied about codon what is the meaning of charging of tRNA? Means amino acid is getting attached on the tRNA, which involves first the activation of amino acid and then the attachment of the activated amino acid to the tRNA. Then we studied about the mechanism of translation initiation. What are the various steps they are formed? First, there is ternary complex. Then ternary complex binds to the 40S to form the 43S complex. 43S converts it into 48S, and 48X complex finally converts into the initiation complex. Then we studied about the elongation process, how elongation takes place, what are the different steps in elongation process. Then termination, which involves cleavage of the growing peptide chain and recycling. Okay, so with this, we have completed our full syllabus from here. So now we will have one more class on day after tomorrow. That is day after tomorrow on 18th April 24. It's just a 1 hour class from 7 to 8 p.m. In that class, we will just try to summarize what all we have studied right from the starting up till end, and we'll try to revise some of the questions that we have solved. We'll just try to revise the questions. Okay, we'll revise the questions, we'll try to solve the questions, and if you have any doubt regarding any of the chapters, you can ask me. Then we'll try to solve your queries in that, and then that would be the final, that would be our last class. So day after tomorrow on 18th of April from 7 to 8 p.m, that would be our last class. Okay, so anybody has any doubts? Any doubt, anybody? Okay, so now I think we'll stop the class now, and yeah, we'll meet next, we'll meet just day after tomorrow. Okay, thank you, Madhvi. Fine. Bye bye, everybody.
Yeah, ma'am.